welcome everyone. It's my great pleasure to introduce our guest speaker today. It's Heidi Rehm from Harvard University from Boston. Heidi is a professor of pathology at Harvard Medical School. He also serves as the chief medical genomics officer at Mass General Hospital. She co-directs the program in medical and population genomics at the Broad Institute, and she also serves as the medical director of the Broad Institute Clinical Sequencing Lab. Her research is focused on discovery of genetic causes for rare conditions, rare human conditions, and she's been very much involved in efforts to improve interpretation of genetic variation through her work with the ClinGen consortium. And she also works now on the integration of genomics into clinical care within internal medicine and more broadly across different disciplines. She holds multiple leadership positions. Just to mention a few, she's a PI of the ClinGen consortium that I already mentioned. She also is a PI of the All of Us sequencing program, where she coordinates sequencing of all of us participants with return of results, a massive effort. She also co-leads the GNOMA genome aggregation effort to improve information on variant frequency, critical information that has been really great for improved interpretation of clinical variation, genetic variation for clinical purposes. She also is a recipient of Kurt Gorshan Award from ASAG. And it's a real pleasure to have her here to talk about advancing genomic medicine through global collaborations today. Thank you.
Thank you very much. It's great to be here and also talk to some of you online. I'm going to talk about a few different projects that we've been involved in. There we go. So one of the themes that I'll talk about a fair bit in the beginning of my talk is around deciphering the causes of rare disease. And one of the challenges that we all face is that, generally over 60% of the cases that we test in the clinical lab remain unsolved after we, perform exome sequencing or genome sequencing. And as a result of that, Daniel MacArthur and I started the Broad Center for Mendelian Genomics and the Rare Genomes Project a number of years ago to try to address this issue. And of course, there's many other programs around the world also trying to solve, all of the causes of rare disease. And there's, many estimates around what are the remaining causes of rare disease. And this is a figure from a paper from a few years ago, identifying those here in gold that we've already solved, ones that have been mapped but unsolved yet, those that we don't think we'll ever see in a real live human because they're probably going to lead to embryonic lethality. But then half of the genome, or really half of the genes in our genome, being undetermined in terms of their role in disease. Now, if we go to the current OMIM website, the current stats instead of 15% is really now right around 24% of genes in our genome have been implicated in a Mendelian disease. And anyone go to the OMIM website and get these stats, which I show here on the left side. But there's also been speculation around, well, how many are we missing? What's left? If we can't diagnose the majority of the patients we test, we must be missing a lot of genes. And what we understand now is from some estimates looking at both our GNOMAD database, where you can look at constraint, so whether genes don't tolerate loss of function, which would suggest that disrupting them will lead to a severe phenotype. You can then, from that constraint, estimate the likelihood they are involved in a rare disease. Or you can look at animal model data, and because there's a lot of similarity of mouse genes to human genes, so if there's a mouse disease, you could speculate that there's a likelihood that that gene disrupted in humans. So when you do that, you get some number, at least 4,000 more genes, estimates up to 10,000. So there's still a lot of work to be done in discovering the causes of rare disease, is sort of the take-home message. Now, one of the challenges, I said, well, 24% of genes have been implicated in rare disease. How real is that number? Do you believe every paper you read that's been published? And we actually have been trying to get a better handle on what is the evidence for each gene and its role in disease, and is it valid? And so we formed this consortium called the GenCC, Gene Curation Coalition. And the first intent was actually to bring some of the public resources together, like OMIM, ClinGen, the Genomics England panel app, Orphanet, et cetera, and get some harmonization between these different resources that catalog genes, roles, in disease. We also later then discovered that there were lots of clinical labs and other platforms. They were also curating gene-disease relationships, particularly as they were trying to decide which genes do I put in a genetic test for a certain indication. You know, a cardiomyopathy panel or a hearing loss panel, they have to curate the genes to figure out which ones to put in. And all that data was not publicly available. So we got all these groups together. We developed consistent terminology for how we characterize the relationship between a gene and disease in terms of evidence level. And then with the goal of really publicly sharing all this information in one consolidated database for the community. And I like to call it the ClinVar for genes. Like you can submit your interpretation of a variant to ClinVar. Well, here you can submit your interpretation of a gene to GenCC. So we launched now maybe a year ago, lost track how long it's been, the GenCC database. It's under search, thegencc.org. We also have a paper and a podcast all about it. And on the right here is our steering committee, which represents members of all the groups contributing data to this database. Just a sense of where we started with this project. These were some of the public resources and the terms they were using to characterize the validity of gene-disease relationships. And you can see that there was a lack of terms in some areas. Terms were different. Some systems use color coding. The two that use that use them in the opposite orientations. Red meant strong evidence in one, red meant low evidence in the other. So it was kind of a mess. So we went through multiple rounds of a Delphi survey process and came up with a set of terms which are almost identical to the ones that ClinGen developed through our framework for gene disease validity assessment. So that's what went into the GenCC database. So everybody now maps or has converted to using these terms for all of their gene disease relationships, with the exception of Orphanet and OMIM don't assess gene disease validity. So all of those entries just go in this new category we had to create called supportive, which, you know, in theory, all of those genes that are in those two databases have some evidence, but they haven't really delineated the level. So this gives you a sense of what's in there. We have 16,911 submissions of gene disease assertions across 4,700 genes from 7 different submitters. So there's lots of data going in, and we're still collecting more and more. And once we have API-based automated submission, we'll be integrating the OMIM database into this resource as well, but they're waiting for us to launch the API. And this is just a screenshot. You can go to the website and look up your favorite gene. I just showed GGB3 here, and you can just click on it and it will expand right in front of you, or you can click the gene and go to a different screen and see a lot more detail. But you can see that, you know, ClinGen has curated this gene for 2 different diseases, both a skin disease and hearing loss, calling one strong, one disputed. Genomics England has called the skin disease strong, and Panel App Australia called the hearing loss limited. So actually it's quite consistent, but you can see it represents different disease claims on a single gene, which is actually quite common. So we have standardized disease terms that we can use, so we know when things are the same or different. So lots of progress in really developing these gene disease resources, but and a lot of the discoveries over the recent years have transitioned to using next-gen sequencing as the mode of discovery. But you can see there's a little bit of a trend here, and this actually kind of continues into more recent years, where it's actually getting harder to make these gene disease discoveries. And one of the reasons for that is the prevalence of each individual rare disease can be quite variable. And this paper from the Orphanet database, what they're basically showing here is that, you know, almost 80% of patients with a rare disease walking around on the planet actually have one of the smaller number, 149, more frequent, more prevalent rare diseases in the range of 1 to 5 per 10,000. So most people with a rare disease actually have one of these more common ones. We've figured out the cause of most of those. What we haven't figured out the causes of often are the really rare ones, where there's over 3,000 rare diseases characterized that affect a very small percentage of the population. These are diseases that are more rare than 1 in a million. So most of the work yet to be done and yet to be discovered is really this end of the spectrum of incredibly rare disorders. And that makes it more and more challenging to actually decipher the cause of rare disease. And there's continued organized efforts. So the Centers for Mendelian Genomics that were funded by NIH in 2015 to 2021 now have evolved into a new consortium called the Gregor Consortium. And the members of that consortium are shown here on the lower right. We are one of the funded groups and really working to apply a lot of different approaches to tackle gene disease discovery, both through global collaboration, strategies around exomes versus genomes, I'll talk a little bit about, and RNA-seq, use of a democratized platform that we developed called Seeker, new methods development, a lot of effort in rapid data sharing, and then functional validation of candidates to prove causality. So this is our team. It's led by Anna Donna-Luria, myself, and Mike Talkowski, with Caitlin and Daniel co-investigators. And then we have a lot of amazing people in our team that are doing all of the hard work at, developing recruiting and curation and analysis and project management, methods development, software engineering, etc. We also have grown a huge consortium of academic partners who are recruiting rare disease patients that we then work with them to sequence, use our platforms and our methods to help solve these cases. But the investigators really lead their own discoveries and the publications that come from them. And so to date, we've over the last, now it's been 7 or 8 years, we have a lot of data, and there's been over 300 well-supported novel gene discoveries from all of these collaborations with our investigators, but still also a lot of candidates still waiting for more evidence to prove causality. And we still remain, at a level of a lot of cases still remain unsolved, where we still need to develop even better approaches to figuring this out. We also, I mentioned the Rare Genomes Project, so this is our direct recruitment arm. And our goal is really to create a diverse rare disease cohort where we could also then be completely in control of widespread data sharing of all that data. When we work with investigators, we're somewhat reliant on their approaches to data sharing. Here, we can absolutely control how widely we want to share this data, which we do very widely. Everything is electronic in terms of recruitment and consent, so we can recruit from all over the US. And in fact, we've been able to recruit from every state and territory with a reasonable diversity of individuals and age range from 1 month to 94 years. At lunch, I was just sharing, we just made a diagnosis of someone who's 94 years old and been on a diagnostic odyssey her whole life. And actually the orange dots are all the participants for whom we've returned a result to them and given them a diagnosis. Despite a lot of these cases going through everything clinically and never getting an answer. And this just gives some statistics around our discovery. So of the cohort we've enrolled, we've been able to give a very clear diagnosis to about 16% of these individuals. And then we have either a strong candidate or a moderate candidate for another subset. And up here plots the age range of the individuals by diagnosis. And what you can see is we're making diagnoses, everything from the young kids on up to people over 50 that have been trying to get a diagnosis for many years. So we continue to solve cases. Now, I mentioned one of our goals is to be able to widely share this data. And one way we've shared that data is through the KG challenges. So we've been able to give individual level data. And I think we put together a set of about 30 some odd cases to be able to give to the computational community developing innovative algorithms to diagnose rare disease cases. And so across the bottom is all the different platforms and algorithms that different groups used in this competition. And we knew the answers and they didn't know the answers. So it was a competition to who would score and make the most diagnoses across this data set. And then we scored all of their answers. And in fact, we even got 2 new diagnoses out of this through some of the methods that the groups had developed. So it's, not only are we sort of crowdsourcing the problem, but we're also helping, because when you develop these platforms, it's very hard to validate them when you don't have large, sets of data to do that with. And then we're going to be running another competition this year. So I'm going to give a couple of examples for this talk of actual cases, particularly our endeavors into thinking about non-coding contributions to genetic disease. So this is a patient with central nuclear myopathy presenting with profound infantile hypotonia, weakness, ptosis, and reflux. They had a strong clinical suspicion for myotubular myopathy, but they were negative for the most common gene involved in that phenotype, MTM1, and nothing found on exome sequencing as well. So we ended up doing trio sequencing and also were able to do RNA-seq on a patient muscle tissue biopsy sample. We applied everything we could in terms of methods and analysis to this case. And ultimately, through the RNA-seq analysis, we were able to see an outlier, very low expression of MTM1, which was in fact the primary gene candidate the clinicians had originally suspected, but saw nothing by routine sequencing. But by using both the outlier expression analysis and looking at exon splicing data, you can see that there's a skipping of an alternative splicing for this particular exon in the MTM1 gene. And then when we dove into the intronic region, we could, in the genome data, we could then see a retrotransposon line 1 element that had inserted into intron 3, and that was actually mosaic in the mother as well. So we were able to solve this case, ultimately using a combination of genome sequencing and transcriptome analysis. Another case is a 6-year-old female with a neurodevelopmental disorder. You see the range of phenotypes here on the left side, so a syndromic presentation. Lots of normal analyses that were done, including clinical exome sequencing by GeneDx in 2017. They reanalyzed that data every year. We then did a research trio genome. We reanalyzed it 3 times in a row and just couldn't come up with any candidates. So a very challenging case. However, we were able to do RNA-seq analysis for this case, and it showed a really interesting sort of set of results that was surprising. So there were actually 37 expression outliers, shown both overexpression and underexpression of lots of different genes. If you compare the number of outliers you see compared, like how often does anybody see this sort of finding on other, analyses? Well, when you plot the number of outliers, this case is way on the, outlier end of the outlier spectrum. So it just seemed abnormal. But in looking at, those genes, we could not find any rare variants that seemed to explain, why these genes were outliers. However, what we did then discover, and this is the work of Vijay and Ben, looking at the fact that there were retained introns, if you look more closely at this data, but not every intron. You can see right here, this particular intron is retained, but not the neighboring introns. And in fact, the over and under expression plots were actually not directly related to the gene being overexpressed, for example, but it just falsely gives you that signal because there's more genomic data from the retained introns that makes it look like it's overexpressed because of that intron retention. And we also think the underexpression may be due to nonsense mediated decay of of certain genes and their transcripts because of the retained introns. So the same intron retention is actually leading to some genes looking overexpressed and some being degraded, was our suspicion. Now, simultaneous to this, one of our analysts, Stephanie, was looking at another cohort, and they had marked a particular gene because of the phenotype for her to look at. She realized that that gene was a non-coding gene that wasn't part of our pipeline. So she ended up looking at cases with this particular gene in mind and discovered an actually known pathogenic variant in ClinVar in this particular gene in the very case that was being analyzed by RNA-seq. And that case not only had a likely path, but also had a novel variant as well. Now, interestingly, because it's a non-coding gene, it's actually misannotated in ClinVar as class 1 because it essentially sits inside an intron of that gene. So you don't even know that this is this RNU 4-ATAC gene. So all those variants get annotated as intronic to class 1. But it turns out this is actually a non-coding RNA gene that operates in the spliceosome. And it's been implicated in multiple different phenotypes, Tal's, Rothman, I forget what these stand for, but a variety of different phenotypes. One of the known variant is shown here in an area where there's a collection of other pathogenic variants. But then our novel variant is out here in this stem region of the RNA gene. So, we're certainly suspicious at this point, particularly because what's known about the function of this gene is that it's part of the U12 dependent minor spliceosome complex, which actually operates on less than 0.5% of introns. About 850 minor introns in 700 genes are affected by this type of splicing, which is exactly the pattern that you saw with that one intron being retained, but the rest not, and only affecting a certain number of genes. So the story really started to make sense. We're now collaborating with a couple labs to functionally prove that novel variant actually has an effect. But we had enough suspicion and evidence at this point to return the result to the family. The case, had a number of overlapping features with other diseases due to this gene, although one of the most prominent features, skeletal dysplasia, was not present in our case. So, it wasn't a home run phenotype match. But interestingly, after we returned the result, one of the unique phenotypes of these disorders is immunological phenotype with IgG and IgA deficiency, lymphocyte B and antibody deficiency. And when the family followed up, they were able to show that all of the vaccinations this child received, there was no antibodies to any of those vaccinations. So it actually matched this other phenotype that they hadn't assessed at the beginning. So at this point, we think this is pretty much a home run diagnosis, although we are still following up with the functional validation of the novel variant. But it just sort of points out that some of these non-coding genes just don't come up in the standard pipelines that people are running, which is why this known pathogenic variant was missed and not ever looked at and things like that. So, so that's a challenge, I think, for our field that needs to really embrace the non-coding genes and other regions. We have found in our hands, this work of Vijay Ganesh, that about 14% of cases, mostly looking at some of the neuromuscular cases where we can get tissue biopsies, have been able to be solved by adding RNA-seq data to those analyses. But we'll still continue to strengthen our approaches in this place. We've recently launched, and the work of Conrad Kachusu was launching our non-coding constraint track in Nomad, so that you can start to see where enhancer elements and other things might have a higher likelihood of influencing gene expression and other traits. It's still not highly resolved, at the base pair level. We're in 1 kb segments at this stage, but with more data over time, we'll be able to get more resolved with that. And this is just a screenshot of the constraint that you can see in the Nomad browser now. And this is work that Siwei Chen and Conrad's group has done. And there's a preprint on bioRxiv. By the way, I think I forgot to mention at the start, I did put my slides on a Google site so that you can download and use them freely. And I'll put the URL up again at the end of the talk. So all of this you'll access if anybody wants to go back to anything. So a lot of people ask, is it worth doing genome if you've already done an exome? What more are you gonna, learn from that? So we've been looking at this, particularly led by Monica Wojcik in the team. And so from 750 families that we've sequenced with genome sequencing, most of them would have actually been solved by a current exome analysis with a current quality of CNV calling on that data. But what we found is about an increase rate of 12% for things that you wouldn't have caught with a current quality exome and, good CNV calling algorithm. And that falls into both structural variants, about 40%, things that are in the small, just like that line 1 element only in an intron, which you wouldn't have picked up from exome CNV analysis, or an inversion or other complex events that you just can't pick up on exome. Also, improved coverage of coding variants because of the capture step in exome, where some regions don't get captured well, and so genome is more even with its coverage. Certainly the deep intronic variants, and that's also helped by RNA-seq. And then it's much easier to look at tandem repeat expansions with genome data than it is with exome. So that's been our experience to date. So our kind of roadmap to continuing to improve diagnoses, obviously more patients, but also focusing on some of the technology. We're doing a lot of work now with long read genome sequencing and more with RNA-seq whenever we can get our hands on tissue, doing more proteomics and functional studies to resolve our candidates, and then incorporating improved reference genome. There are still missing genes in the current genome build where the sort of T to T assembly is now uncovering some of these genes that we can start to look at in our patients. Also, I'll talk in just a second about really trying to democratize rare disease analysis through our Seeker platform and doing more data sharing. So I mentioned the Seeker platform. So this is an application originally started by in Mark Daly and Daniel MacArthur's lab. Daniel and I then really built it up when we started the Center for Mendelian Genomics. It's an open source software platform to do exome and genome analysis. We've now installed it on the Anvil platform, so anybody can bring your data and put it in a Google bucket for yourself, and you can use this platform to analyze it. It's FSMA compliant, so it's all the security requirements. And we now have over 2,000 people from over 100 countries that are performing analysis on this platform. We continue to add new features because it's open source. Some groups have developed their own modules that we've then pushed into the common primary platform. And we're continuing to push out new features. So if you're looking for a free platform, come see us. And we've even created YouTube tutorials on how to use the platform from our analysts that do a great job recording these. It really encourages the community to work together because you have to constantly innovate with new technologies and techniques and methods for analysis to keep these platforms alive and innovating. It also allows more standardized sharing of metadata so that as we share our data, when we export it out of Seeker, it comes with all the rich annotations that both are put on both through our pipelines as well as through our analysts that are annotating. I looked at this is a good candidate, bad candidate, all the different things that we annotate both automated and manually. Now, I mentioned this earlier that so many of the rare diseases are incredibly rare, and it will absolutely, the only way to solve these diseases is going to be through data sharing. Early on, the more common rare diseases, an individual investigator could amass enough cases themselves to just publish on their own, but not with these really, really rare diseases. So I'll just give you, one of many classic examples. Rare Genomes Project case 434, 2 SIBs, microcephaly, seizures, global developmental delay, non-ambulatory. So we sequence, this family, we find 2 novel variants in the PPIL1 gene, which had never been linked to disease, one from mom, one from dad. One was a stop codon, which is why it looked more suspicious when we saw it, but the other was just a routine missense variant, not enough information to certainly implicate this gene in the patient's or the family's phenotype. So this would be a classic example. All right, let's throw it into Matchmaker Exchange and see what we get. So the problem here early on was people would come across these candidates and there's nothing they could do with them. So they literally sat on the shelf sometimes for decades with no matches, no other cases. And so a number of years ago, a lot of us got together in the rare disease community, recognizing that this was a serious problem, and said, okay, how can we solve this? We just need a simple system to do what we then coined genomic matchmaking. And so if 2 groups had a candidate gene designated from looking at a case like I just showed you, you could just put it into a system and match. And all you need is like gene D matches gene D, right? And then you could also enter the phenotypic features and look for overlap. And we actually, each could calculate semantic similarity scores from HPO terms to say what the phenotype similarity was as well. So we actually, this was a concept we came up with, and we began building a solution starting at a meeting we had at ASHG in 2013. We defined the goal. We also defined the barriers. The barrier was paranoia of getting scooped. Everybody had their like prized candidate gene that they wanted to publish on, and were not willing to like stick it out on a website. So when we built the solution, we kept that in mind. And the solution was nobody can see the candidates. And you only see somebody's candidate if you have the same candidate and you actually match. And then both investigators get emailed simultaneously on the same email. So it's very transparent. You both have a case. Here are your genes. Here are your phenotypes. Now collaborate. And so it was like a safe way of sharing your data. And so we built all the infrastructure. We connected the first 3 databases in 2014. And then began building out the platform, which we called Matchmaker Exchange. So that family that I just showed you, we stuck it in Matchmaker Exchange, and we immediately matched with 7 other families shown here. So ours was family 8. It was already actually a collection that had been building in Matchmaker Exchange, and Joe Gleason was leading the paper and even had made a mouse model at that point. So we actually solved that case immediately by sticking it in Matchmaker Exchange, because this was already underway with matching. But this, sometimes you start the match and other times you end the match, but it's an incredibly powerful platform. We now have 8 rare disease databases that are nodes around the world in many different countries. There's actually over 15,000 candidate genes from over 200,000 individuals from 13,000 contributors from 103 countries. And that just is a testament to how powerful it is that everybody is using it. In the recent publications that we put out in Human Mutation, there's over 1,000 novel genes that have been discovered using the federated platform here. So really, really important platform. But this only works if everybody's found their candidate in their case, right? And sometimes you look at a case, I already told you, like 60% of the time we can't solve these cases. You look around and you see nothing interesting. So we have to solve those too. And this platform doesn't help if you haven't found a candidate to put into the platform. So we have come up with this concept of, the 2-sided matching is what we do today. Each group has a candidate, 2 sides, you come together. But what about 1-sided? You have a candidate and you just you're like, I'll bet somebody else has this variant or this type of variant somewhere in one of their cases. And you want to query their raw data for a match. So that is what we are now working on. And of course, ultimately, we want to do the 0-sided matching. Nobody finds any candidates, throw all the data together and look for commonalities. But that will take us a little more time to do that globally. So we're focused on this 1-sided matching. There's some of the groups within the Matchmaker Exchange have actually launched individual interfaces to their data where you can actually query their raw data. So this is just a screenshot of me registering my username in Variant Matcher. And then I put in my search, I found this variant with these phenotypes in one case. And what I get back is a list of the 8 cases in their database who have that variant and all of their phenotypes, which I show here in red, which includes 4 patients with rare disease that have all different phenotypes, and then 4 patients who are unaffected parents. So I was able to quickly rule this out as a candidate, which is equally as important as ruling things in, is to get rid of things and move on. So this is, showing some of the power of being able to query more deeply, even though, nobody had marked this as a candidate in any of those cases, and rightly so, because it wasn't a good candidate. So we are, forming, now a consortium to really start to work and bring groups in to create a new federated platform that will actually enable querying raw data. We're working with the Global Alliance and some of the new APIs that they've developed that have more flexibility, because we not only want to query, like, does any of your case have this variant? We also want to query, is there a loss of function variant in your case in this gene? Because often when we do gene matching, it's actually not the same variant. Almost never actually is the same variant. It's actually a different loss of function or a different missense. So we can say, do you have this type of variant in this gene in your raw data? So lots of things to come. Now, interestingly, although we've been focused on rare disease gene discovery, we all know that every time you get a genetic test for known genes, you get lots of VUSs back. And so this platform will also be really critical for VUS interpretation also in the genes we know today. And in fact, if you look at ClinVar, the largest fraction of variants in ClinVar are over here in yellow that are either VUS, everybody agrees they're uncertain, or they're conflicting. So either way, they're uncertain, and we need to resolve these variants. So bringing together more data sets where the data is phenotyped and we can get access to the phenotype of that data will be powerful. And that just falls into a larger effort within ClinGen of really trying to better interpret variants accurately. And now there's no way that expert panels in ClinGen will classify all of the billions of variants in our genomes. There's no amount of centuries of work that can do that. But we take what I call the four-pronged approach. We develop common standards, so everybody doing classification is actually doing it the same way. We get everybody to share their data in ClinVar so we can see the evidence, the rationale, and what you said about a variant, and then bring people together to share more data because you saw who actually saw it. We resolve conflicts between labs, and then we, for the hardest variants, we focus on the expert panels resolving those variants. Now, we did a little project recently looking at the rate of uncertainty in genetic testing. And the original reason I did this study was I had heard insurers say, we don't want to pay for exome and genome sequencing because it creates too much uncertainty. All those VUSs and those genomes and exomes are just going to lead to downstream healthcare costs. So we're not paying for exomes and genomes. And my suspicion was, I think the problem is worse for panels. So we thought we could answer this question. Let's just get all the data from all the clinical labs and ask the question, how often do you report VUSs in panels versus exomes and genomes? Simple question. So we gathered data from 19 labs from the period of 2020 to 2021. There's 1.5 million data points from what you see as the large labs doing all this testing. Now, 97% of that was from panels. That's what most of is happening today, in part because insurance doesn't cover the genomes and exomes very well. But statistically significant, it was undoubtedly clear that panels generate more uncertainty than exomes and genomes, and I'll explain why in a sec. But either way you look at it, 30% of all genetic tests are inconclusive due to VUSs, so emphasizing the core issue at hand. Now, not surprisingly, the rate of VUSs goes up with the size of the test, obviously. But then the paradox here is, then why is genome less, right? So the reason is just around laboratory practice. So when we do panels, it's always the assumption, well, the physician ordered the panel because the patient has that phenotype that matches all the indication for those genes. So it's important to report the VUSs because they are in genes related to the phenotype, and then the physician can follow up with studies because there's a high probability. That approach made sense when you were doing small gene panels with high prior probability of pathogenicity. It gets worse argument as you get to these massive over 200 gene panels, where the prior probability that any VUS in those genes is actually pathogenic goes way, way down. But when you move to genome, you generate thousands of VUSs. Yes, that is true. But we can't put thousands of VUSs on a report, right? So what do? We ask the physician for detailed phenotype. We look carefully at all those variants and the genes they're in, and we choose carefully which variants to put on the report, which is only ones that have a very high suspicion for a role in disease. That is why the rate of VUSs is much lower in genomes and exomes, is because of practice and professional interpretation. So you should pay the geneticist. Now, we interestingly, many years ago in a lab that I used to direct, we developed sub-tiers of VUSs, the ones that were very suspicious for a pathogenic role, the ones that were very suspicious for being benign, and then the rest in the middle. And you can see how they divide here across those different categories. Quest, Athena also have been doing this, as well as Baylor. So some labs have taken this strategy. And what we found is, although overall, if you look at all VUSs, the majority get downgraded to benign over time, and only a quarter of them go up. But then if you look at the sub-tiering, the likelihood variants go up is much higher for the VUS favor pathogenic, and this is data from the LMM. Also, the data from Quest and Athena were published, and here they show from their sub-tiering, the higher tiers also much more likely to move to pathogenic compared to the ones that are the low VUSs. So really emphasizing that we could make use of these structures to reduce the amount of uncertainty on reports. So the new guideline that we're working on right now, we're actually going to subdivide the VUS landscape into 3 different tiers. And then it'll be directly related to the point scoring system. So it'll be automatically delineated. And then we are working on another work group that I'm working on forming that will give guidance for how to use these categories in terms of reporting on reports to really work to reduce the amount of uncertainty going back to patients is the ultimate goal. I will note, I put down here, the UK actually has had sub-tier guidelines for a long time, and their 6 sub-tiers are labeled hot, warm, tepid, cold, ice, and ice, cool, cold, and ice cold. Although these do say don't put these terms on a clinical report, but they use them internally. So we're not the only ones that figured out that this would be useful. So anyway, in summary for this piece, there are certainly benefits to putting VUSs on reports. It allows follow-up and tracking of these variants when there's a higher suspicion that they could convert to pathogenic. But we really need strategies to reduce how many VUSs are going back, and we'll need guidelines, which we're now going to work on. So the very last piece that I'll just talk about in my role as Chief Genomics Officer at Mass General Hospital, I formed this genomic medicine unit, a small clinical team that's really been trying to address the integration of genomics into medical practice. And so we've been engaging stakeholders, identifying pain points in the process, and trying to deliver creative solutions. This is the current team we have doing this work right now. But I formed what I call the Genomic Medicine Implementation Team, which is representatives, both physician and, where available, genetic counselor representatives of every clinical division. The blue boxes are from Mass General, where I started this, but now I've been working to add Brigham Women's Hospital and other hospitals across the Mass General Brigham ecosystem. And so we all come together quarterly to really discuss strategies and solutions. I'll do things like polling, like what's your biggest pain point? The last poll, epic integration of genetic test results and other issues. So we've been really trying to tackle the issues that our physicians have with integrating genetics into clinical care. Yes. Sure. You want to take a screenshot?
The one before, yeah.
Yes. All of, and you're welcome. As I said, you can download the slides. The URL is on the last slide. You can just, there's even a barcode. You can use your phone to scan it. So yes. So one, it's because top of the list was epic integration. We actually started looking at all our send out data to different clinical labs, interviewing all the providers. Why do you order from Invitae or GeneDx or Quest or whatever? What were the important things? What were the issues? And then we developed a list of preferred versus acceptable versus discouraged laboratories across the full list of laboratories. And then that allowed us to prioritize the integration efforts, which aren't simple for integrating with the EHR, by you starting with the top preferred and going down from there. And now we're working on these integrations. We also created a genetic counseling network and deployable genetic counseling services. So the kidney group wasn't using genetics at all at the time. We provide on-demand genetic counselors and just assisting them in getting test ordering set up. And now they're using it robustly across the clinic. Andrew Lundquist actually even wrote a paper about the utility of this and getting this clinic set up. And then got educated enough that he no longer needed the genetic counselor, and now is just using a genetic counseling assistant with the things he needs. But we also have this network. They meet monthly. They bring together common issues across genetic counseling. Things like we created a common consent form across all of the healthcare system, got it approved, and now we have, because of so much telehealth, and a big pain point was getting the patient to physically sign a consent form when they're remote. They've now integrated electronic signatures, just simple things that can make a big difference in deploying genetic services. We also heard, from our cancer clinics, they were inundated by at-risk cancer patients. These are patients who don't have cancer, but meet NCCN guidelines for being at risk. And the cancer clinic could not see the hundreds of patients that were coming through this clinic. Also, preconception carrier screening, a huge increase in demand, couldn't be handled by the OB/GYN department. So we started to try to address this sort of gap between primary care and specialty clinics with this preventive genomics clinic service. And most, our target was not the wealthy people who wanted their genome sequenced. It was these populations being underserved. And that was in fact our largest, set of patients coming in, mostly referred from primary care. But the challenge was we only have so many physicians doing this clinic. So we've now just starting next week have switched the service, which is now going to be a genetic counseling fast track service, where we actually have the genetic counselors work directly with primary care. Primary care physician signs off on the genetic test order. So our clinicians in the PGC don't have to see the patients. And that will streamline this process and allow us to see even more patients and really begin to engage primary care in a bigger way in the delivery of genomic services, but which for routine things like cancer panel screening and preconception screening have to transition to primary care at the level they're being expanded on. We also launched an e-consult service so that physicians can just ask questions, we can give answers, and that also helps educate them. And we rarely get a second question the same, from the same physician, because once we answer it once, then they know how to deal with that in future cases. And we've actually been tracking the e-consults, looking at outcomes. So what did we recommend? Then looking at the medical record to see if they actually did what we recommended. So these are, the different outcomes of these e-consults. They're mostly being requested by primary care, which is our target for this largely. And out of the 88 actionable recommendations, 78% were followed through by the provider, 17% not, and 5% actually left the system, so we couldn't track. But largely they are taking our advice and following through, which sometimes is discouraging genetic testing and saying this patient's not appropriate, or you should test a different family member, or things like that. So lots of different good advice going back. And I'm going to stop there so we have time for questions. I did want to acknowledge lots of people in my team that do a lot of the work that I talk about. And also I mentioned you can download a copy of my slides if you want them. So there is something in the chat. Happy to take questions. Could you expand upon how the PCP integration works in practice? Is it an interested and engaged group of PCPs or available to any PCPs? So that's a good question. We are giving like 4 talks to all the PCP practices, but this fast track service we're piloting with one particular practice. So it's usually by geographic location, because we come in, we sit down with the PCPs, we give a talk about the service that we're doing to try to engage them for the preventive genomics clinic and e-consult service. But we're also trying to target less, the more diverse clinics so that we can deliver care, because we're finding those in a lot of the diverse clinics, they never get to specialty services. So it's partly based on which practices are most engaged, but we're using leadership to get us into those clinical practices, giving talks about our services, and then that usually leads to more engagement. So yes.
Question?
Yes, go ahead.
So I'm not a scientist at all, so please.
You're still allowed to ask a question.
Yeah. So number one, systems thinking, that's what I'm working on. And the idea of convergence by Philip Sharp. I think this is a very good example of that here. But how prevalent is it in the industry, the pharma business? 2, polygenetics. I know that it's prevalent in heights, phenotypes, height, and skin color and stuff, and some people talking about intelligence being like polygenetic. How hard is it to track that? And the last one, rare diseases, they're not profitable for the pharma industry, or I don't know, the CRISPR is used, because when it's very rare,
Mhm.
it is a problem. So how do you solve those issues?
Um,
Too many questions.
Yeah, that's a lot of questions. No, but I'll go backwards because I can remember the most recent one first. So we are working with an organization called Rare is One, which has been funded by the Chan Zuckerberg Initiative, and they're really working with patient advocacy groups to help them sort of work with pharma and biotech in terms of therapeutic development, teaching them how to interact. And our group is also funded as part of that project for a different reason. But one of the projects we're working on is a project we call the prevalence calculator. And we actually use our Nomad database. So far, we've just been doing it for rare diseases because you can estimate prevalence by carrier status in the general population. Because one of the critical things to engage pharma and biotech is to be able to say, what is the prevalence of your condition, which is not so straightforward. And the more frequent the disease, the more likely they are to engage. We can also even tell them what places around the world have higher incidences based on geographical or ancestry, population differences, things like that. So, years ago, nobody touched rare diseases. I will say I've seen a change in industry with a lot more groups being able to tackle, because the likelihood of response is higher when you target a known target with, a thoughtful approach to the therapeutic, right? So it's a higher likelihood of success, but it is much more cost per patient because you have a smaller population you're targeting. So, there's definitely been a lot of activity in the space. Whether it's going to tackle all, 7,000 or more rare diseases, that remains to be seen. It will have to get to places where common approaches can extend across multiple diseases to reduce the clinical trial costs and everything else, probably to be able to tackle more rare diseases. But I think there's promise. So I'm going to go back to your next. I'm going to see if there's another question here. I'm going to switch to one of those, and then I'm going to come to your second one, just democratizing questions. Given the low zero rates of GC reimbursement, how are funding the additional GC and GC efforts across your institution? So this is a challenge that we have been experiencing. So in the beginning, the low volume clinics, I had enough genetic, a few genetic counselors that could just help out. Now what we're doing is we're actually getting, so like the next genetic counselor I'm about to hire, because the cardiology practice needed a 50% FTE. So then I email my genomic medicine implementation team. I say, anybody need any fraction of an effort? I just got 6 responses. I could use 5% here. I could use 10% here. So they're funding those fractions, but they don't have to commit to a full hire, right? So that we are still needing to have, I just can't provide it for free from my unit. I don't have infinite funding, but by, organizing it, we can spread out those costs and be more efficient with the resource. So actually, that's not your question. That's whoever asked me here. Now going back to your second question, which was PRS scores, I think it was, and you had said you've seen them in height and other things. There's actually polygenic risk scores, right? So there's lots of polygenic risk scores. In fact, we were just talking about Christoph's work in calculating them for polycystic kidney disease, but these are being developed across many different phenotypes: cancer, cardiovascular disease, obesity, diabetes. The eMERGE consortium is now, I think there's 10 different polygenic risk scores. We're running all of the genotyping arrays at the Broad, and then I sign out all the PRS scores that then go back to patients in the eMERGE consortium, to all, thousands of patients. And then the eMERGE consortium is studying how these results are used. So to answer, I think what your question was, yes, there are PRS scores for lots of different phenotypes that are actually starting to show more clinical utility than they have in the past in terms of actual utility. And really what I would say is spreading the population to decide who to treat more aggressively versus less aggressively. There's still controversy as to how useful, but they're becoming more useful, I would argue. And then I completely forgot your first question. Something around.
I was just thinking that my idea was like, how do you build trust, like global trust when you do collaboration?
Yes, carefully and strategically, I would say, are the key words. But it really is about engagement, understanding what it is you need to solve. Now, people are going to come to the table if it's an issue they care about, but then you have to develop solutions that keep so like the matchmaker exchange, our solution wasn't, all right, everybody give us you give your data to us and then we'll manage this process. No, that's not going to work. So we developed it. Everybody's data stays in their own database in their own country. They control access to it and they control how to share it. And then we, this whole paranoia about getting scooped by developing a solution where everybody got notified immediately at the same time, got people over that. So you have to think about the trust issues, what people are, how to balance sharing versus control and other things, and really making sure you come up with solutions that address the key need, but also are cognizant of people not wanting to just turn things over to somebody else. So I think that's a key issue.
Thank you very much.
Sure.
I'm sorry for asking.
No, I don't see any others in the thing, and I think we might be out of time. So.
I'll ask maybe
Yes,
one more question. Within the scope of your genomic medicine activities at MGH, is there room for pharmacogenomics, and have you do you have any efforts aimed at implementation of pharmacogenomics? Yeah,
it's interesting. So when I was first meeting with each of the divisions and asking them how they use genetics today, because I wanted to get a sense of the problems and challenges. One of the questions some of them said, well, we ordered this pharmacogenomic test. And I said, oh, you're ordering just one pharmacogenomic marker for, X, treatment that you encounter. I said, why might you not order like a full panel? Because then if the patient, it's the same cost, patient gets a different drug later, you'll have it all ready. And the answer was, I don't want the liability of dealing with those other results that were ordered by me. And so like in my mind, pharmacogenomics is only going to be useful if those results are incorporated in the electronic health record with clinical decision support on top of it. So the physician goes to order a drug, who's not the physician who ordered the test, that information comes up. And I can say Epic, has some of that functionality. It's not robust, but other institutions have built slightly better systems, Vanderbilt, some others out there. So I think that solution needs to be more robust. And I discovered that Mass General Brigham did not buy the genomics module with Epic, so we don't even have access to it. Now we're changing that. But these are problems, real problems, to actually implementing the robust use of pharmacogenomics. And we do have pharmacogenomics clinics that are doing this for people that request it, but it's not routinely being offered except in certain, high use scenarios, like back of ear and, other things. But
we have the genomics module, but we don't know how to use it.
Well, and it's not perfect. The people who have it that I've spoken to, they're like, yeah, it's hard.
What you're doing is wonderful. The problem is the genetics community who does all these tests, they know how to use them. Physicians, many of which are in the 40s or 50s, have no minimal genetic knowledge. Yep. How are you teaching them? How are you educating them about how to use these genetic tests? Yep. Not to be afraid of them.
No, it's a great question, and that is why we've targeted primary care so much in these endeavors. So we did just work with our the Harvard Catalyst to develop a course that's a CME accredited course, so they get CME credits to learn about genetics. So we launched that. But I don't feel that's the best way to educate them. I really feel real time, patient shows up in the clinic and they need to deal with it. They're motivated and willing at that moment in time. And that's why, launching this e-consult service so they could quickly ask a question and we can get back to them. And now working with them to provide the genetic counselor with the primary care person to integrate in their clinics. So that the genetic counselor is doing the pre-test counseling, filling out the genetic test order. And then when the results come back, they're actually explaining the results. If there's follow-up, they're helping with that. But I have found, in a lot of the that example with Andrew Lundquist, he didn't know how to navigate in the beginning, but then we, taught him how to do it. And now he does it most on his own, is teaching his nurse how to do the ordering and is more on his own. And so I think over time, by helping do this with them, we will educate more of them in how to embrace this, but it's not straightforward.
As a matter of fact, the genetic counselors are many things, but the most valuable things is they're really educators.
Yes,
and
absolutely.
If there was some way in the letter, rather than just say this is what you should do.
Mhm.
Paragraph explaining why you should do it, what's behind it, may go a long way in educating people on capabilities.
Yeah, and we are definitely doing that like with the e-consult service in terms of when they ask a question, it's not like, we'll just do this. It's, well, why are you doing this? What's the rationale? So we are doing that in these e-consult services, educating the reasons for it. And that's why we rarely see repeat questions from the same providers. In fact, we never have, because we do feel they're learning from the answers to questions and the way we explain it. But it's, we're not going to solve all the thousands of primary care that, should be helping embrace genetics. It will take time. All right, well, thank you, everyone online.